Good afternoon, ladies and gentlemen. My name is Sume McPhail. I'm the founder and CEO of WEQUIL School, a project-based, self-directed virtual learning platform. WEQUIL's mission is to help all people reach escape velocity, the point where your natural strengths are leveraged to provide service to others. At WEQUIL School, all students are teachers because teaching is the best way to learn. We create projects that solve real problems because that is the best way to grow. We share them with you because that is the best way to build ourselves and our relationships. All parents and young people are free to collaborate with us to help achieve a mission. For more information, please go to wequil.school. Today's presentation on genomics leverages the unique strengths of our students to serve you by sharing key insights into genomics technology and to help you understand its vast implications for healthcare, agriculture, the environment, and ethics. What is genomics? Genomics is the study of the human genome and how it works, what it is made of, how it has evolved, and mapping it out. Genomics has become very relevant today because of two reasons. Costs in genome sequencing has declined from many thousands of dollars just a few years ago to less than $300 today. This is leading to an exponential rise in adoption in data on our genes as well as relationships to diseases and other health outcomes. 5 years ago, scientists stumbled upon a technology they call CRISPR-Cas that enables us to edit genes. This has been curing people of genetic diseases and could very well be one of the most important breakthroughs in healthcare. I'm going to pass it over to Seth Pellig, who is a mentor for WEQUIL School.
Thank you, Sume. My name is Seth Pellig, and I'll be sharing my article on genetically modified babies. Although this may seem like a joke, humanity actually did this. Dr. He genetically edited two Chinese twins, possibly preventing them from ever getting HIV. The doctor did this through a gene editing technology called CRISPR-Cas9. CRISPR remembers a virus, and when it detects it, it sends out RNA segments to find its DNA. Then it cuts it out. In this case, Dr. He used this to remove a gene called CCR5, which he then artificially induced a summer version back into the twins. This is the first time that anyone has ever changed human genes in a way that can be passed down throughout generations. If this experiment succeeds, it can open up infinite possibilities. We may be able to make ourselves immune to any disease, infection, or virus. We also might be able to go even further and make ourselves stronger, smarter, and live longer. Although it may have major impacts, genetic modification on humans is seen as off limits worldwide. It is also illegal in places including China. Knowing this, Dr. He carried out his experiments in secret, and this led him to be sentenced to 3 years in prison. All in all, we have already changed our DNA, a thing many people call playing God. It is a major responsibility that could also result in countless unforeseen consequences. Therefore, this is a discussion that must be fully debated until we can recognize it as a reality.
Thank you, Seth. Now Anya is going to present her project on the Human Genome Project. Anya has done a lot of great stuff on history, and I'm very excited to hear her thoughts on the Human Genome Project.
Thank you, Sume. I'm Anya, and today I will be presenting the Human Genome Project. It was a 13-year-long research project initiated in 1990 to determine the exact makeup of human DNA. The human genome is a complete set of genetic instructions and material needed to build each person and allow them to grow and develop. A genome is made of your chromosomes, DNA, and DNA bases. The project's key objective was to determine the sequence of the material in all human genes. All the hard work of thousands of scientists from all over the world came to a successful conclusion in 2003 when they published the complete sequence of the human genome. Because of the Human Genome Project, individual human genomes can be decoded on a computer for only $1,000. Such sequencing can help identify genetic abnormalities and help with further research. Most importantly, because of the Human Genome Project, the COVID vaccine was developed.
Thank you, Anya. Now, Isla is going to present her project on genomics miracles.
Hello, my name is Isla McPhail and I am 9 years old. Today I will be presenting an article that I wrote about genomics miracles. The first miracle is how genomics cured sickle cell. Sickle cell turns red blood cells into a sickle shape. Red blood cells carry oxygen into different parts of your body. So when you have sickle cell, you're not able to get enough oxygen. The first person who was cured from sickle cell by gene editing was named Victoria Gray. When she got cured, she was feeling major impacts. She was feeling so much better that just 1 year later, she felt good enough to go on her first airplane ride to visit her husband. Next is COVID-19. COVID-19, just 1 year ago, the COVID-19 lockdown happened. When we all went on lockdown because even though you can only die from COVID-19, you may only likely die from COVID-19 if you are older or already have some sort of sickness. It is very contagious. Usually it takes 5 to 10 years to develop and test to develop a vaccine and test it. With genome sequencing, it only took 2 days to develop a vaccine and 1 year to do all the testing. Lastly, there's a disease called spinal muscular atrophy, for short, SMA. SMA causes weak muscles in infants that are born with this disease. One girl, the weak muscles get weaker and weaker until they eventually do not work. This affects when babies learn how to crawl, swallow, and even breathe. One girl who had it was named Evelyn. When she got cured, she was showing major improvements. When she turned 3, you could never even guess she had SMA. Another girl was named Sophie. They saw improvements as early as 3 weeks after she got cured. For conclusion, genomics is actually making miracles happen.
Thank you, Isla. Now we know that genomics is doing many things, but how does it actually work? DNA, as Anya already mentioned, is the set of instructions for an organism to live. That includes humans. Genetic diseases are caused when there is a mutation in your DNA. Gene sequencing has allowed us to identify these mutations early, and most importantly, the costs have drastically dropped, making it so that many more people can get their genes sequenced. We have been influencing genes in plants and animals through selective breeding, but not until CRISPR-Cas was developed have we actually been able to directly edit genes. So how does CRISPR work? CRISPR is made up of RNA and a type of protein, either Cas9, Cas3, or Cas12. RNA is similar to DNA, except it's only one strand and doesn't have the double helix. Using RNA and mixed with Cas9, Cas3, or Cas12, the RNA can identify which DNA you want to edit, and the protein can actually edit the gene. Aqua Bounty is a company that is genetically modifying salmon to make them grow faster and need less food. Their salmon are able to grow 30% faster and only need 70% of the food. This same technique can be applied and can have major impacts on agriculture. There have been many experiments using CRISPR to see what can this technology do and pushing the boundaries and possibilities. For example, there was a dog who had their genes edited to make it so that there was a 300% increase in its muscle mass. There was also a cat that had its gene edited to glow by taking a gene in a glowing jellyfish. The question is no longer, can we do these things? This is already happening and is already a reality. But the most important question now is, what should we do and what are the potential consequences? Cadence will be covering this in his article on the dangers of gene editing.
Thank you, Sume. Gene editing can be used for good things like everyone else mentioned, but it can also have consequences such as replacing genetic diseases with new ones. Editing DNA is a technology that has been very popular over the last 2 years when Chinese scientist Dr. Hu edited human DNA for the first time using CRISPR, a new gene editing technology. Today, I will be going over the history of DNA editing, the social consequences of editing genes, and the possible unforeseen consequences of changing DNA. There are benefits and risks to gene editing. I hope this article can help you rethink how we should approach gene editing in a way that allows the benefits. Humans have been impacting their genes forever by means of natural selection, which is basically just evolving naturally without any artificial at all. And we've been changing the genes of plants and animals by means of selective breeding, which is taking two plants or animals of the same species and with wanted traits and breeding them together. An example of this with plants is broccoli, cauliflower, and kale, which was created through selective breeding from the exact same plant in the mustard family. An example with this is dogs. We kept breeding the fastest dogs until we got the greyhound, which can go at speeds to up to 40 miles per hour. There are many social consequences when DNA editing goes out to the public. Countries that do not support gene editing might start fighting countries that do. People may move to another country that does support gene editing, pushing those countries who do not support gene editing to support it, or the exact opposite. The military could also make super soldiers, which may cause a World War III, like in movies Wrath and Khan or Allegiant. There are many bad things that could happen after we edit the genes, like accidentally causing other genetic diseases like cancer, diabetes, and maybe even undiscovered ones. Some genes even influence our personality, so how do we know that we're not changing who someone is? And what are the consequences socially when we change someone's physical appearance? There are many different consequences that could happen after. Gene editing will have irreversible consequences. We may not be able to go back if World War III happens or if entire species go away out of existence. Today we talked about the history of gene editing, the social consequences, and the unforeseen consequences. I hope this presentation changed the view on what you think about gene editing.
Thank you everybody for presenting your projects on genomics. If anyone watching wants to learn more about WEQUIL School, you can go to wequil.school, which is also where you can find all the projects that were presented today.